So let's see a show of hands. How many of you have ever had problems finding your car in a busy parking lot like that? Okay, fair number. Now I noticed my kids didn't raise their hands. When you're young, you don't have a problem finding your car. I mean, maybe you're not allowed to drive. But if I mention this to my students, they don't know what we're talking about. So if they drive into this parking lot, they have their music on, they talk to their friends, they go into the shopping mall, they come out a few hours later and they walk straight to the car and drive off. And they never think, where did I park my car? Because the brain, this amazing supercomputer that we heard yesterday from Henry and others, is just able to make these what we call spatial maps, ingrain them in our brain, at least for a short period of time, so when you need them, they come back immediately. And you don't even have to think about it. But as we get older, this becomes harder. And with people who have Alzheimer's disease, they can't even form these maps and they can't retrieve them. Now, we're actually not the only species who have that problem. Mice, monkeys, even dogs, they have the same issues. As they get older, their brain doesn't function as well, and they have problems making and retrieving spatial memories. So what happens to this brain, to this amazing functioning brain that when you're young, there's just all these amazing things, and when you get older, it doesn't work as well? And even if you're in your 50s and 60s, you start to notice that sometimes a word doesn't come immediately, or you forgot something that you wanted to do, and it never happened to you when you were young. So we know that as we get older, the connections, the synapses between neurons, the nerve cells start to disappear, they start to die off, the cells actually start to die, and the wiring starts to disappear, so that a 90-year-old person has lost roughly 10%, or 100 gram, not 10%, 100 gram of the total mass of the brain. Now we heard yesterday, it doesn't matter how much weight you have, how much your brain weighs, but if the connections are lost and the wiring is lost, that of course makes a difference. And the brain becomes susceptible to age-related diseases such as Alzheimer's. And estimates, or numbers actually, from many different populations show that at age 85, up to 30% or 40% have Alzheimer's disease. So in the US, there's about 5.5 million worldwide, maybe up to 50 million people have Alzheimer's disease. And we know this disease starts early. It starts 10, 20 years before you have clinical symptoms. Now, unfortunately, we can't do anything about it. So this is really doom and gloom, if you will. There's nothing that we can do at this point. And the fastest growing populations in the world are actually the above 75-year-olds across the globe, not just in the developed countries. But there will be more and more people, and society will not be able to afford to care for all these people. So we really need to have an understanding of what happens with this brain as it gets older, and we have to fix that problem. Now, it's hard to study the brain because obviously you can't take a biopsy from the brain, and all we know, or most of what we know about the brain at the molecular level, is from people who have died and donated their brains. And then we can study them at the molecular level, or we know it from animals. But most animals we study, such as mice, are not quite as sophisticated, of course, as humans are. But what we do know is that the brain is connected to the rest of the body. And the whole body, of course, ages. So we know as we get older, all kinds of age-related diseases start to creep in, such as arthritis, heart disease, and as well as other age-related diseases. And you can see here, of course, the obvious sign, the skin starts to change. But so what we need to understand is how the aging body interacts with the brain. And maybe if we are able to do that, we can start to study the rest of the body, and we don't really have to get access in living people to the brain, but we can, and you will see later on how we are trying to do that. So what can we do about this? Wouldn't it be great if there was a fountain of youth? This magical idea that people had in every culture, every tradition, every society. The Chinese had Shifu, who was sent by the Emperor Qin to try to find the elixir of life, the fountain of youth. Alexander the Great spent a lot of time trying to find the fountain of youth, and Ponce de Leon, who was sent from Europe to find it, spent much of his time sailing the Caribbean to find this magical water that they thought would give you eternal life and would keep you healthy and young forever. This image here, or this picture here, is from Lucas Cranach the Elder, and it shows how old people enter this fountain on the left-hand side and they re-emerge young and rejuvenated on the other side. Now, unfortunately, I have to tell you, we don't have a fountain of youth yet, but what's really exciting is that, at least in mice, there seems to be a possibility that there is something like a fountain of youth. There is a revolution that happened over the past 10 years or so in the aging field that allowed us to slow down aging and even reverse aspects of aging. And this is with a model that is called Parabiosis. This was actually this model was invented over 100 years ago in France. It's somewhat creepy. And it's similar to surgically generated Siamese twins, if you will. So what scientists did is they make a cut in the flank of a mouse or two mice, and they suture them together. And specifically, that's why I said it's a bit creepy. Specifically, what Tom Rando, a friend of mine who has actually his lab right to mine, together with Irv Weissman and Irina Conboy, when she was a fellow in his lab, asked the simple question, why does a muscle get old and doesn't regenerate when it's injured in an old person or in an old animal, but it does so when you're young? And he asked the simple question, is it because the stem cell in the muscle lacks something inside the cell that makes them old and makes them incapable of producing young muscle tissue, or is it something in the environment that is responsible for that? And to ask that question, he took an old mouse and he paired it with a young mouse. And in this model, the blood vessels start to fuse together so that you have now a joint circulation of the blood. And so this allowed him to ask, is the blood that comes from the young mouse able to reactivate and restore the activity of the old muscle stem cells? And that's what indeed he found. Otherwise, I wouldn't be standing here. And he didn't only find rejuvenating effects on the muscle, but he also noticed effects in the liver. And he saw that in the brain there may be increased stem cell activity. And that's how we started to work together and then really explored that in more detail. In the meantime, other scientists have shown that you can regenerate and rejuvenate parts of the pancreas, the kidney, the heart, bone, and maybe more. People are still working on that. And we're now 4 or 5 labs who can show beneficial effects on the brain. Now, the blood is connecting all the different tissues in our body. And that's maybe why it has this magical connotation to it. Because we know if you lose the blood, you die, right? But the blood doesn't just carry blood cells that transport oxygen and fight or fight diseases, but it also contains hundreds or thousands of different molecules, proteins that carry information from one tissue to another, from the muscle, for example, to the brain. If you exercise your muscle, that's good for your brain. We know that some of this is carried by protein factors that are able to transfer this information. So the blood, if we study the blood, this may allow us to get an understanding of the brain, because the brain is highly vascularized and uses a lot of blood. So you would think that if the brain doesn't work well, or if it gets older, we may see changes in the blood. And that's indeed what we studied. So we studied in healthy people and in healthy young people, healthy old people, what the composition of the blood is and how that changes as people get older or as mice get older. And what we found that there are dramatic changes. And some of these changes we can actually fine-tune, and those are potentially the ones that are responsible for these rejuvenating effects that I'll show you in a minute. But just to summarize these effects, so what we see, we don't actually have to suture the mice together. We discovered that you can simply collect the blood, the plasma, the liquid fraction of the blood, from one mouse, or from many mice actually, from young mice, and transfer it to old mice. Now this is not quite trivial, but it's doable. So we collect maybe blood from 50 mice or so, we get rid of the cell, and we inject it repeatedly every 3 days, a small amount into old mice, and what we notice is that these old mice have now increased stem cell activity in their brains, they have less inflammation, their nerve cells communicate better again, we can measure their electrical activity, and it looks more like a younger mouse. And most importantly, their memory function is improved. And so how do we test that? This is a parking lot test for a mouse, if you will. So this is called the Barnes maze, and what we do here is we have a table that is about 1 meter wide, 1 meter above the ground, and has a lot of holes. And we teach the mouse to find the single hole where we mount a tube underneath. Now the mouse hates this environment because there's a lot of bright light shining on it. We even have a fan that blows wind on it. And the mouse really doesn't like an open space because that's dangerous. So we can teach it to use the spatial cues, such as this checkerboard, the triangle, the rectangle, to make a spatial map. Like we humans on that parking lot, we would use a lamppost, the storefront, and our brain makes a spatial map. So the mice are able to make this map when they're young. And when they're old, they have a problem doing that and retrieving it. So we teach the mice usually 4 times per day, 4 trials, 4 days in a row. And if the mice are young, they're actually able to do this. But when they're old, they look like this mouse here. They're lost. So the red arrow points to the hole where the mouse could escape. But these old mice can't really find that hole. They can't perform this task. But if we give the same age mouse young plasma, either from young mice or even from young humans, we found that you can use plasma from young humans. We infuse it into these mice every day, every 3rd day, a small amount, about 5%, 5 to 7% of the total volume of blood of the mice. And look what these mice are able to do afterwards. Almost looks like the strategy is a little bit different. The mouse just doesn't start walking, but it's like, where am I? Now, this was the best mouse we ever had. So if you look at that statistically, you actually find that these old mice make up maybe 30% of what a young mouse would do, 30 to 50%. It depends on the experiment. And we can reproduce these. People are blinded to do these tests. They don't know which ones are treated, which ones are not treated. And we get reproducible effects here. We also were able to find some of the factors that might be responsible for this. But because this is so easy, plasma is actually given to people. If you have an accident, if you have to go to the hospital, you have a surgery, plasma is given to millions of people every year. So it's a very safe treatment, and I was encouraged by neurologists. I'm not a doctor myself, but I was encouraged by neurologists to try this in humans. So we started a company, everybody does that in Silicon Valley, and did a very small trial to see is this safe in old people. And we treated 18 patients with mild to moderate Alzheimer's disease. We gave them 1 unit of plasma per day, per week, 4 weeks in a row. So they got a total of 4 units. That's about the same ratio that we gave the mice. It's about 5% of the total blood volume. And then we did standard assessment. Now, this is a very, very small trial. So we were not able to see any of the functional benefits. But we have confidence that it's safe. We didn't see any unexpected side effects. And so now we moved on to a bigger, larger so-called phase 2 trial where we test different doses. And the patients who receive it, they actually don't know whether they receive a saline solution or whether they will receive the actual plasma. And the plasma comes from young people. So what do you think about this? Should we stop this? Should we continue this type of research? Are there ethical concerns here? What if this works? Would we have enough plasma to treat everybody? Probably not. Could we maybe recruit more people if you knew you're a young person and you knew this could help your grandma who has Alzheimer's disease? Maybe they could be able to recruit it, but it's unlikely that we would have enough. Because I mentioned to you also, this may not even work, not just work for the brain, but it may work for other tissues as well. The muscle, as Tom Rando has shown, or the liver, or the kidney, and so forth. So we really need to understand what this goes. But what I also wanted to share with you, when we published these results, there was a lot of interest. And there's potentially bad things that could happen with this. This is an example. Silicon Valley is a show on HBO. I don't know, has anybody seen it? Maybe. So here the protagonist is a Silicon Valley CEO, and he has a blood boy who gives him young blood. So this came out of our research. They talk actually about parabiosis and say they want to use the young blood so that he is energetic and can be an efficient CEO. But even worse, this company here, Ambrosia, initially they had all our papers on their website, and I'm not sure if you can read it, but here they say young blood is an exciting new therapy which shows promise for aging and the reversal of conditions such as Alzheimer's disease. We never showed that. We said in mice that are old, we can restore some of their cognitive function, we can reduce inflammation, we can activate stem cells, but we didn't say this works in people. We have to try this, we have to be serious about it. But people can misconstrue that, and they can try to make money with it. You pay, for 1 liter, you pay $8,000. Obviously, clearly not a good way, a direction of how. So if we ask these questions again, we should probably stop this type of behavior, right? And again, if this works, maybe we want to make it available, but we have to figure out, does it really work? Does it work in blinded trials so that we can be confident? But ultimately, really, we need to understand, how does it work? So if we have, again, this body, the brain, that is this amazing machine, this supercomputer that has this amazing capacity to learn and to build the world that we have. And that can go bad as we get older, that we completely lose our identity. We don't know anymore who we are, probably, and we certainly don't know anybody around us anymore. So how does it work that this young blood can potentially have beneficial effects, at least in animals, but possibly also in humans? What are the factors? I mentioned to you, we have identified some of the bad factors, so I didn't tell you that the young mouse in this pair of young and old mice actually suffers from the exposure to old. It shows completely the opposite effects that the old mouse. Everything the old mouse benefits, the young mouse suffers from. They have less stem cell activity, they have more inflammation, they have less neuronal activity, and their memory gets impaired. That allows us potentially to find bad factors associated with aging that we could block, and we have identified a couple of them. One of them is actually entering now a phase 2 trial where we try to specifically block this factor with a small molecule. So what are the factors? We need to get a better understanding of this. Where do they come from? We don't know which tissue in the body makes these factors. If we knew that, maybe we could target that tissue. When you get older, we could try to keep that tissue younger. Which cell types does young blood rejuvenate in the brain? We don't know yet what is the actual target in the brain. Is it the blood vessels? Is it the glial cells, the support cells of the neurons, or is it the neurons themselves who benefit? We think actually it's all of them. When we look at the gene expression level, when we look what are the genes that get activated in different cells, we see that all of them show some changes in response to young blood. And that brings us to another very fascinating question with aging. Do all the organs in our body age at the same speed? We don't know that. Is it possible that some of our organs age faster than others? And if these organs produce bad factors, maybe then they cause aging of other tissues. So again, if we understood that, we could start interfering there. And then lastly, as I just mentioned, it's possible that this aging could then spread through the body. And there's some exciting developments that show that there are cells in our body, they're called senescent cells, that are not forming tumors, like some cells do when they get old and they get sort of out of control. But these cells are unable to divide, but they don't die. They don't get removed by the body. So they just sit there and they produce bad factors. And what people have shown is you can specifically kill these cells in mice and you again have rejuvenating and beneficial effects. So a lot of people are trying to target now these bad cells in the body, get rid of them, so that you can again have beneficial effects. So where does this all lead? What is the future in this? Is there going to be a brave new world where some people get young plasma, others don't? Where we will have selective treatments for certain types of diseases but not for others? And what I think is going to happen is there's going to be a paradigm shift in medicine, and we're in the middle of this right now, I think. So in the Renaissance, alchemists started to get an understanding that there are different elements in the world, that there's potentially even ways to interfere with the physiology, with disease in our body. The concept of drugs and poison was established by Paracelsus, for example. And in the last couple of centuries, physicians started to analyze the body more carefully, and they started to notice how different organs work. Cellular theory was invented by Virchow. We started to recognize there are individual cell types in an organism that form organs, and we could figure out what are these organs doing. We could get an understanding at the physiological level how these different organs in our body work with each other. And over the past 40, 50 years or so, we went ever deeper in trying to understand the molecular machinery of all of this. We figured out, to a large extent, how the individual units, the cells, work, how they communicate with each other. We started to understand the blueprint, the DNA of the whole organism, and even the modifications of the DNA, which is called epigenetics, how this all encodes to generate these amazing organisms that we have. We started to understand individual proteins. We can crystallize them. We can understand their structure. We can use that to rationally design drugs that interfere with the function of these. But what that also led to is that we went ever deeper and deeper in trying to understand the minute details, and to some extent we lost the big picture, how this all connects with each other. But over the past few years, an amazing revolution has taken place with new technologies coming in and computer science helping us to look at all this data together and to really put this knowledge that we have generated over the past few decades at the molecular level into physiology again. And I think we will see over the next 10, 20 years a revolution in medicine, in part because we will have an understanding of how these molecules work together and have a molecular physiology. So if we go back then quickly at how I led on with the talk, if you live 80 years today, and this is the average life expectancy in a lot of the developed countries, most people will start showing signs of frailty and disease at age 60 or so. 50 to 60, the first signs of aging are to creep in. And by age 75 or so, people take 5, sometimes even 10 different drugs to treat all these different ailments. And I show this with this orange color there. But what we're going to do, I think, over the next 10, 20 years, is to push this disease back so that we can extend the healthy lifespan, that health span will be extended. And I think we start having the tools to do this. And I think this is not unreasonable, because there are actually people living right now that do not get sick. They die without getting sick. Now, this sounds a bit like an oxymoron, but centenarians, they live 100 years, and many of them have no diseases. Now, they're not running around like a spring chicken, but their body does not suffer from heart disease, cardiovascular disease, neurodegeneration, diabetes, arthritis. They don't have these diseases. They frequently are healthy. They get sick a few months, a year, and then they die. So that suggests that it's possible to have our human body be healthy for a long period of time. And at least if you live 80 years, that you may be able to enjoy 70 or even 75 years of health. I think that will be the future. And maybe as a consequence of that, we will extend lifespan as well. People will get older. But that's not really the main focus. Really what we want to try to produce is that intermediate bar here, where people can live longer, can live healthy for a longer time. So should we try to extend lifespan? That's one of the questions that always comes up. What is an ideal lifespan for humans? Now, people live increasingly up to 100 years. Should we live longer? What would be the consequences if we all of a sudden would live to 150 years? What would be the social consequences? What would be the consequences on population growth? With that, I leave you. Thank you very much.